Hello and welcome to today's webinar on tissue engineering of the respiratory epithelium. My name is Anja Lüngen. I am a PhD student working at the Department of Biohybrid and Medical Textiles in Aachen, Germany. As cell culture with primary respiratory epithelial cells is part of my daily lab work, I am really happy to share my experience with you and to give you some insights in culture methods and applications. I'd like to start with a short introduction in who we are and what we do. The Department of Biohybrid and Medical Textiles, or Biotex for short, is part of the Institute of Applied Medical Engineering at the RWTH Aachen University in Germany. Although the institute is part of the University Hospital in Aachen, we are located in a different building, the Center for Biohybrid Medical Systems. At Biotex, we develop tissue engineering solutions with a clinical focus on cardiovascular, respiratory, and heart tissue engineering implants, for example, vascular grafts or biohybrid heart valves. Two of these three working groups are located in Aachen, and the third working group on heart tissue engineering is located in Maastricht, the Netherlands. As tissue engineering is a very interdisciplinary field, all working groups at Biotex are influenced by different research fields: biomaterials research, textile engineering, bioreactor systems, and of course, as the major goal of all our engineered constructs, the translation into the clinic. But let's now start with the respiratory epithelium. In vivo, the respiratory epithelium acts as a very specialized tissue that covers most parts of the nasopharynx, the trachea, and the bronchi. It has a pseudostratified columnar structure and consists of three major cell types that are polarized. Near the basal membrane, the basal cells are located that represent the precursor cells for the other two cell types. Most cells are ciliated cells. That means that they carry specific organelles on their surface, the cilia. These cilia can beat actively, and by this movement they transport the mucus that is produced by the third cell type, the goblet cells. This self-cleaning mechanism of mucus production and cilia transport in the airways is called mucociliary clearance. It provides a first defense mechanism against pathogens and pollutants. In a periodic acid Schiff reaction or pass reaction, the mucus in the goblet cells can be visualized by a pinkish color. The cilia can be found on the epithelial surface using scanning electron microscopy. In vitro models of the respiratory epithelium have gained more and more influence in the past. Their value in studying respiratory diseases such as COPD or asthma was recognized long before the COVID-19 pandemic. These models make it possible to investigate the pathological mechanisms and facilitate the finding of new treatment options. An important aspect lies, of course, in the possibility to use human tissue, which on one hand provides more reliable information and on the other hand meets the efforts of the 3 R's principle to reduce animal experiments to a necessary minimum. But respiratory in vitro models are more than just a disease modeling platform. They may also be the treatment option themselves. In tissue engineering, researchers aim to develop tailor-made replacements based on patient-derived tissues to overcome limitations regarding donor numbers or immunosuppression in cancer patients. Finding the ideal combination of cell type, scaffold, and in vitro conditioning may in the future enable the development of individual replacement tissues. With respect to the airways, maintaining the mucociliary phenotype represents a key issue in this context. In vitro culture of respiratory epithelial cells is divided in two phases. In the initial proliferation phase, the cells are cultured under submerged conditions in order to receive a confluent layer. Successful differentiation requires formation of an epithelial barrier that is dependent on confluency. Therefore, the duration of the proliferation phase varies between different labs and different protocols, as it is also dependent on cell source and seeding density. The epithelial barrier formation can be monitored by measuring the transepithelial electrical resistance, or TEER. After an initial peak, the TEER decreases and remains constant. In the second differentiation phase, the apical medium is removed and the cells are brought into direct contact with the air. This phase can last for several weeks. And to achieve these air-liquid interface conditions, or ally conditions, the cells are seeded on specific cell culture inserts that are available with different membranes and different pore sizes. Usually, a pore size of 0.4 micrometers is used. As we will see on the next slides, the medium used in both phases also plays an important role, as the medium demands differ between the phases. Besides the medium, there are some other aspects that influence the differentiation success of epithelial in vitro cultures. The cell source plays an important role. Primary cells can be of nasal, tracheal, or bronchial origin, and usually they are most similar to the in vivo situation. Nevertheless, the cell behavior may differ between nasal and bronchial cells, and also between different species. As primary cells only provide a very limited lifespan and also require connection to the clinic to receive the tissue for cell isolation, researchers also use immortalized cell lines or tumor-derived cell lines. The phenotype of the latter, the tumor-derived cell lines, is not really comparable to healthy tissue. But literature reports mucociliary differentiation of immortalized respiratory epithelial cells. Still, their use is not recommended for tissue engineering applications due to their genetic modification. Next to the cell source, age and medical history as well as the lifestyle of the donor influence the differentiation outcome. The cell passage and the seeding density should also be taken into consideration, and as already said, the medium plays a major role. This includes the use of antibiotics and specific growth factors like, for example, epidermal growth factor, EGF, or retinoic acid, but also the medium volumes and the exchange frequency. Last but not least, the choice of the insert membrane and the insert coating before cell seeding impacts the cell behavior. In the past few years, various media for the proliferation and also the differentiation phase have emerged on the market. In our study published earlier this year, we wanted to evaluate the suitability of 4 different differentiation media to raise a mucociliary phenotype in our experimental setup, using primary human nasal epithelial cells isolated from surgical samples and used in passage 1. Our setup consists of Corning inserts with a PET membrane, a human collagen 4 coating, and special deep well plates called thin-sert plates that allow larger basal medium volumes up to 4 milliliters. In the proliferation phase, we used airway epithelial cell growth medium, or AECGM, from PromoCell for all samples. In the differentiation phase, we compared the 4 commercially available differentiation media. Modified airway medium was made of AECGM and DMEM. Pneumacooled ally medium and mucil air medium are ready-to-use media that are available on the market. And EMM was a mixture previously published by our institute consisting of endothelial cell growth medium 2 and mucil air medium. The first electron microscopy analysis of our inserts revealed cilia formation in 5 out of 7 modified airway cultures, while pneumacooled and mucil air led to cilia formation in only 1 out of 7 donors. In EMM, 2 out of 7 donors developed cilia. On the slide, you can see representative pictures of the scanning electron microscopy. Basal bodies and the characteristic 9+2 configuration of the microtubuli inside the cilia could be observed under the transmission electron microscope for ciliated samples. 10 independent and well-educated observers scored the cilia amount on all samples in a blinded manner. This qualitative scoring procedure showed significant differences between the 4 media, with modified airway medium and EMM to be the most promising media regarding cilia formation. Histological analysis via a periodic acid Schiff reaction confirmed presence of glycogens, mucopolysaccharides, and glycoproteins in modified airway cultures, indicating mucus production. For the other media, cell monolayers were most abundant. Again, representative pictures are shown. Immunohistological analysis was performed with antibodies against 4 different epithelial markers. As can be seen in the native control on the left, alpha-tubulin staining confirmed presence of cilia. Claudin-1 was used as a marker for barrier formation, and mucin 5AC staining showed mucus production, while pan-cytokeratin staining showed presence of a broad range of cytokeratins as a less specialized epithelial marker. Most modified airway cultures were positive for all 4 markers and showed staining patterns comparative to native tissue. For the other media, most cultures were only positive for tight junction marker Claudin-1 and for pan-cytokeratin. We also analyzed the production and consumption of several growth factors using an enzyme-linked immunosorbent assay, or ELISA for short. To begin with retinoic acid, we could observe that retinoic acid content in used modified airway medium after the media exchange was rather low in comparison to the other media. If we normalize these results on cell-free medium, we observed highest retinoic acid consumption in modified airway medium and in EMM. Nevertheless, modified airway medium revealed to be the medium with the lowest retinoic acid content of only 50 nanomolar. Literature supports our findings, as retinoic acid concentrations higher than 100 nanomolar are thought to decrease the differentiation capacity. We further analyzed 8 additional growth factors and found significant differences for vascular endothelial growth factor, or VEGF, epidermal growth factor, EGF, and fibroblast growth factor beta, or FGF-beta. For VEGF, we observed a production in 3 of the analyzed media, with the concentrations being highest in modified airway medium and EMM. This indicates the importance of vessel formation for the epithelial differentiation process. EGF was consumed in all media, while FGF-beta showed varying results for the different media. Literature reports an ideal EGF concentration of 10 nanograms per microliter, which supports our findings. In addition, regulatory interactions between retinoic acid and VEGF have been reported in the literature. To analyze the donor-dependent impact on growth factor content, we compared growth factor concentrations of donors with and without ciliation. VEGF and EGF concentrations were rather low in non-ciliated donors in comparison with samples that showed ciliation. This could be a hint that non-ciliated donors were not able to maintain a certain growth factor level in the medium, possibly because of a decreased metabolism or a lower total cell count due to poor proliferation. The effect could also be observed for FGF-beta in modified airway medium, but not in EMM, probably due to the higher initial FGF-beta concentration in EMM. Retinoic acid content did not differ between ciliated and non-ciliated donors. To sum up our findings, we found modified airway medium to be the most promising regarding the mucociliary differentiation outcome in primary nasal epithelial cells. One major aspect seems to be the balance between the growth factors EGF, VEGF, FGF-beta and retinoic acid, although further research has to be done to evaluate the ideal balance between these growth factor concentrations. Of course, we cannot avoid mentioning the discrepancy regarding the lack of differentiation in pneumocooled ally medium and mucil air medium to divergent reports in the literature. These media have been successfully used in other studies. In the end, we cannot answer this question, but we think that the following aspects contribute to the different outcomes. The cell source, including nasal or bronchial origin, may play a major role, also in combination with differing isolation procedures. As we used AECGM as proliferation medium for all samples, it is possible that we would have seen differing results when using proliferation media made for mucil air or pneumocooled ally medium. The combination of proliferation and differentiation medium may be crucial in this context. Last but not least, we have to consider the unknown medical history, the age, and the lifestyle of the cell donors. As we in our lab usually use surgical samples from patients in the clinic, it might be that these donors exhibit a more complex precondition than healthy volunteers used in other studies. But let's now come to some applications of respiratory epithelial cell culture in tissue engineering. In an earlier study published by our group in 2019, a 3D model of the tracheal mucosa has been developed by combining nasal epithelial cells, nasal fibroblasts, and human umbilical vein endothelial cells, or HUVECs for short. Fibroblasts and HUVECs have been embedded in different hydrogels, and epithelial cells have been seated on top of the hydrogels after polymerization. These tree cultures have been cultured in Ally for 28 days with EMM as differentiation medium. Path reaction showed an almost native respiratory epithelium using a fibrin-based hydrogel, and electron microscopy revealed a multiciliated cell surface. In this study, our group could also show correlations between epithelial differentiation and enhanced vascularization in the tree culture models. EMM was found to be superior for both epithelial differentiation and vascularization in this context, as shown by these 2-photon laser scanning microscopy images after a CD31 staining. The combination of EMM with fibrin-based hydrogels was found to be superior for both vascularization and epithelial differentiation. Immunohistochemistry was positive for epithelial cell markers alpha-tubulin, mucin 5AC, and pan-cytokeratin. We conclude that in co-culture systems, supporting cell types can overcome medium limitations by providing necessary growth factors. Another application in respiratory tissue engineering is represented in one of our projects where we develop a new biohybrid airway stand to optimize the treatment options for tracheal stenosis in cancer patients. With commonly used silicone stents, the tracheal mucosa is covered by the stent, preventing a functional mucociliary clearance. This causes the formation of mucus plugs below the stent and highly increases the risk for complications. The biohybrid airway stent developed by our group has a different concept. The stent consists of a self-expanding nitinol stent that is covered by a polymer cover. This cover acts as a barrier against tumor ingrowth from outside and additionally functions as the growth area for a tissue-engineered respiratory epithelium that maintains the mucus transport throughout the whole inner surface of the stent. To achieve a uniform distribution of cells on the inner stent surface, the cells are sprayed on the stent after stent implantation via a specialized cell spraying procedure using a catheter. In our studies listed below, our group could show that this spraying procedure does not affect the cell survival or the mucociliary differentiation, as pass reactions did not differ between sprayed samples and non-sprayed controls. In addition, the cells developed cilia and were positive for claudin-1 and pan-cytokeratin staining after cell spraying and ally culture. With that, I'd like to finish my presentation and to thank you all for your participation. Special thanks are also made to all our funding agencies and to our cooperation partners who made this work possible, and also to PromoCell for the opportunity to present our results. Now I am happy to answer your questions as far as I can, so please feel free to ask. If you are further interested in the field of tissue engineering, please join the community at the Termes World Conference in November 2021 in Maastricht, the Netherlands. I hope to see you there. Goodbye.